We're moving on to the session, which is a continuation, really, of what we did before lunch, opportunities for collaboration across cohorts, with three presentations that are going to speak about environmental information, nutritional information, and multi-ethnic data. And then we'll have time for some discussion. Jeremy and I are going to try to moderate this, although people were wondering, do you know how to chair a session, Francis? Well, we'll find out. So, first up, David Hunter is going to speak about the value of environmental information and how to gather it. David.
Well, I'd like to thank the organizers for asking me to speak about this topic, and I'd also like them to thank them for asking Walt Willett to speak about diet, because between the two of us, I think we'll be able to give you a view of the importance of collecting environmental and lifestyle information. So, of course, the fundamental importance is we know that the major causes of disease are environment and lifestyle. Just very briefly, how do we know that? Rates of diseases vary tenfold around the world for most of the major diseases. This is global burden of disease, differences in the prevalence of ischemic stroke, and tenfold difference between high and low countries. For cancer, same thing. This is stomach cancer. Men, women, rates of stomach cancer, and stomach cancer is fairly rare in more developed countries. And the opposite is true for colorectal cancer. Again, more than a tenfold variation for both men and women across countries and regions, and more developed regions having higher rates. So it's a perfectly reasonable hypothesis 50 years ago to wonder whether there are country-specific genetic determinants, whether continental ancestry differences in the susceptibility to these diseases is behind these huge differences. But we've known from the migrant studies for over 50 years now that for most diseases, not all, but for most diseases, that's not the case. Because migrants from a high incidence country to a low incidence country within 1 or 2 generations adopt the incidence rates of the new country. They bring their genes with them, but they experience rapid changes in incidence. So over here, these are Japanese rates, first and second generation Japanese in Hawaii, and European ancestry, residents of Hawaii, and within 2 generations, a very rapid reduction in stomach cancer incidence. Even more astonishing, these data are from the 70s. Rates in Japan are much higher now, which again, they haven't changed their genes, but they changed their lifestyle, and now they have some of the highest rates of colorectal cancer in the world. But people moving from Japan almost immediately within a generation acquired the rates of colorectal cancer of people who'd been in Hawaii for a long period of time. I think actually that must mean the microbiome is operating here, because I can't think of anything that would change that fast within a generation. So difference in rates of most diseases between countries and over time within countries due to differences in environmental and, quote, lifestyle factors, not genetic differences. But the revelation over the last decade from the genome-wide association studies is that we finally have, beyond the major genes, very robust information that genotype is also very important within a country. So these are data from the Breast and Prostate Cancer Cohort Consortium on the lifetime risk of breast cancer for a polygenic risk score, which at this point had about the first 80 SNPs. We're now up to about 180 variants robustly associated with breast cancer. And this line here is the 95th percentile. This line here is the 5th percentile for the polygenic risk score. And there's about a 4, almost 5-fold variation in lifetime risk associated with being at the higher end of having inherited a bad hand of these low penetrance alleles versus being at the luckier end. So within countries, most diseases are due to differences in both genetic and environmental and lifestyle factors. We need to measure both. So Walt and I are going to talk about how we measure the environmental and lifestyle factors. What factors are we talking about? Well, these are data from the global burden of disease, attempting to, for all diseases, establish what the major causes of those diseases are globally. So we have conditions like hypertension, pathophysiologic conditions like high blood glucose. Obviously, these have their own both genetic and lifestyle causes. But pretty much all of the other things within the boxes are behaviors or environmental exposures. About half of them are diet. Walt's going to talk about those. And I don't think we need really address smoking at this point, but I'll show some data on how we measure alcohol, BMI, air pollution, and physical activity, just as examples of the differences in risk that we can establish robustly based on mostly self-reported information, often complexed with other databases. So we're all appropriately skeptical about the information people and patients give to us. We're taught that as human beings we're kind of unreliable informants, and so we should be always very, very careful about the type of information we're collecting, its quality, its validity, and the reasons that people are given for its skepticism about self-reported information on exposures. People lie, people just don't know. Many exposures are very complex, so we can't really estimate them, but maybe if we can get a gizmo to estimate them, we'll be in better shape. And then more recently, just in the last couple of years, all of the above, but Mendelian randomization is the solution to all of these problems. People lie about their drinking. They do. But that doesn't stop us being able to get robust information about the relationship between alcohol intake and a variety of diseases. So this is from the Danish Cancer Diet and Health Cohort. Pretty much linear association between increasing drinks per week in men and women and the outcome of liver cirrhosis. So even though individual measurements may not be perfect or maybe far from perfect, we can still get a good aggregate assessment of the influence of, in this case, alcohol intake on this particular outcome. People lie about their weight. So here are data from the big collaboration that John Dinesh and Walt and Richard and others put together on BMI and mortality, the sort of J-shaped curve. And this is for all of the studies that had actually been able to measure weight and height at baseline, clinic visit, visit in person, or whatever. And so J-shaped relationship. Now let's see what happens when we ask people about their weight and height. Essentially exactly the same relation. So even though we know in comparison to measured weight that most of us underestimate our weight by a few pounds, and actually the more obese, the more pounds is the underestimate, self-reported data, far, far less expensive than measured data, unless it's being measured for a routine purpose, gives us exactly the same answer as the measured data. Now, something like air pollution, very few of us can accurately describe our PM 2.5 exposure. I actually just went and checked, and in Durham today it's 27 particles per billion, and the question is, how do we get information when people can't give us the information? So here's data from a cohort of 62 million Medicare participants in the United States, where Joel Schwartz and Francesca Domenici and others essentially asked people, or looked in the database, where did they live, and then was able to overlay the information about, in this case, average concentration of PM 2.5, and then relate that to mortality. And so we're somewhere in here, I guess. So it actually, even though it looks very kind of rural, there is some bad stuff in the air in North Carolina. But when they looked at the relationship between PM 2.5 and mortality, This is just the small piece of the distribution at the lower end of the curve. You know, if China, Beijing or Shanghai today was what we'd be looking at, we'd be in the next room. So this is just up to 10 micrograms per cubic meter. And linear association, even at the lowest dose, so there is no safe dose of PM 2.5, and this was very, very compatible with previous studies that had been done in India and China at literally order of magnitude higher doses. So we ask people, where do you live? Or we have it in a database. We bring in another database, and we can get reasonably accurate estimates of something that's as complex as average PM 2.5 over a year. Physical activity, famously difficult. There's physical activity, there's physical inactivity, there's low intensity, there's moderate intensity, there's vigorous intensity activity. And so this is another consortium activity. This is 6 cohorts with about 660,000 people and about 100,000 deaths over about 20 years. And with this hint of a little uptick at the end, pretty much an inverse association between increased physical activity and lower risk of mortality. And again, building in a lag to account for pre-existing conditions, excluding people with pre-existing conditions, doing a latency analysis. The results were pretty much the same. So more physical activity associated with lower mortality doesn't mean it's causal, but let's have a look at what the accelerometer gives us, and these studies are only just starting to come on stream. So this is 6,000 women in the US Women's Health Initiative who wore an accelerometer for a week, and essentially the same association. I think it's fair to say, Rory showed some data that the accelerometer actually does give you a somewhat better measure than self-reported activity, probably because it's really picking up activity 24 hours a day, not just the things we ask on questionnaires, which are usually leisure time activity, activity commuting if you walk or ride a bike, and occupational activity. But essentially, the self-reported data gave us essentially the same answer as the accelerometer data. It's very, very nice to have both. It's also very, very nice to have accelerometer data on 100,000 UKB participants, because then you can use that to calibrate your self-reported data on 500,000 against the 100,000 that you have the accelerometer data for. So I hope I've shown you that we can get information that is sufficiently robust if the studies are large enough, et cetera, on a wide range of lifestyle and environmental factors. Mendelian randomization is a fabulous technique. John briefly referred to it. I think it's really optimal when you've got a biomarker you're trying to predict and you're curious about whether that's causal, but it's also now being used to look at the genetic predictors of behaviors, but it doesn't always give you the right answer. So here are data from the consortium project, pooling project of prospective studies. These are quite old data. This is increasing BMI in women. This curve is the risk of breast cancer premenopausally, and this curve is risk of breast cancer postmenopausally. So BMI, obesity, actually is substantially inversely associated with premenopausal breast cancer and substantially positively associated with postmenopausal breast cancer. We have mechanisms for that, at least for the postmenopausal association. And the question is, what answer do we get from the MR? So in the BPC3 BCAC analysis, 80,000 cases of breast cancer, looking at the Mendelian randomization instrument for BMI, established from the GIANT consortium with about 200 SNPs, we get the right answer for premenopausal, but the wrong answer for postmenopausal. So if you had only this information, you'd conclude that being obese was protective against breast cancer over a woman's entire life course, and we wouldn't know that actually that's not the case for postmenopausal breast cancer. We can probably explain that. but it just is a warning that it's terrific when Mendelian randomization actually is concordant with all of the other sources of information, but if it's the only source of information, it may or may not be correct. Finally, we've been talking a lot about how we put all these data together on scale, and these are data from Denmark. Somebody referred earlier to the privilege of living in Scandinavian countries with all of the data and databases. Recent publication based on 1.8 women followed for 11 years, and oral contraceptive information from prescription databases. And the previous data, this is the relative risk for current use of OCs, mainly from the first and second generation of OCs. This was the collaborative study that Valerie Beryl led. Relative risk of 1.2-ish. Here's the data from the Nurses' Health study. This was mainly second generation and starting to be third generation assays, hormones. 1.3, 1.2 for the current third generation assays, oral contraceptives. The point here is that we need to be monitoring these things, and once we have an association, it's important to see if it persists. There was a lot of hope that the more recent contraceptives would not be associated with risk of breast cancer, but it turns out that they are. Gene-environment interactions, quickly, this is actually good news. When we started the whole story of gene-environment interactions, we were looking for associations that would be super synergistic or cancel each other out, that would be beyond multiplicative or beneath multiplicative. But when you look for most diseases, this happens to be breast cancer, the polygenic risk score and the lifestyle information is independent. And so if you're at a twofold risk of genetic and twofold risk of lifestyle, that's a fourfold risk overall. This greatly simplifies the risk modeling. Here's data from Lanjan Chatterjee and others from the BBC Three, looking at 10 deciles of polymorphic risk score for breast cancer, and then looking at absolute risk of breast cancer according to high versus low risk associated with non-modifiable or lifestyle risk factors. And the genetic risk score gives you a nice increasing in association, but the relative risks for the lifestyle factors are about the same, 2 or 3-fold, high to low, in each decile of risk. The difference is that there's a much larger number of cases associated with adverse lifestyle risk factors for breast cancer if you're at high genetic risk than there is if you're at low genetic risk. So it may be very important to target in diseases people who are at high risk because they are particularly susceptible, but it's not because of some synergy. It's just because if you're at higher, you start off at higher risk and you have a doubling of risk, that's going to be more cases than if you start off at lower risk. And SEC has shown the same thing for heart disease, low, medium, and high genetic risk on the PRS, and this is unfavorable lifestyle and favorable lifestyle. Relative risk is about 2 in each category of genetic risk, but there's a lot more excess cases here if you start off at high genetic risk. So this is good. It doesn't mean that the interaction term in the logistic regression for the interaction is giving you a significant p-value. In fact, it's not. It means that we can take the lifestyle information, and with a very few exceptions, particularly in the sort of drug idiosyncrasy world, environment and lifestyle just models. So the key is, even though we'd like to have genetics and environment in exactly the same studies, we've got to get the environment right and the genetics right, and if we do that in independent studies, that's probably going to be okay to give us risk prediction. Just finally, all of the problems associated with cohort studies. We've learned a lot over the last 20 years about how to mitigate those problems. We've heard about regression dilution and calibration approaches to deal with measurement error. MR, when it works, can help with confounding. Latency analysis, we heard, very important. If there's any reverse causation, this continues to be a problem. I think the biggest problem is false positives due to people analyzing small, underpowered studies. And that's something that, as a group in consortia, we can do. Just to point out that if you're interested in harmonizing your questionnaires, when Francis was the director of NHGRI, he funded a group not too far from here to come up with a Phoenix project, and online you can get state-of-the-art questionnaires now in multiple languages that if we all used, we'd have much more poolable data. And then just to finish, we started cohort studies with interviews. We had thousands of people. We moved to paper questionnaires, often optically scanned. Now, clearly, we're using digital questionnaires, touchscreen in UKB, online, mobile, integrating apps, and that should allow us to get information from tens of millions of people. And then if we can follow people in a system such as we heard of today, we're planning this in the UK in a series of studies. There's got to be an acronym, right? Hoping to get information from about 5 million people that can be followed through all of the mechanisms that Kathy Sudlow showed us. Then maybe with, that'd be cheap. Maybe with about 5-fold of funding, we could ask them to spit in a cup, and about 10 to 15-fold of funding, we might actually get a blood sample from some people. But the key is to get environmental and lifestyle information from as many people as possible to get it right. I've said all of this just to reinforce that we need large sample sizes, particularly in the precision medicine era, when we're now making one disease into 5 or 10 diseases. And this can be obtained in large population-based studies, integrating information, and if we have interoperable EMRs or system-wide coding, then we can start to do even bigger and hopefully better studies. And just to thank a lot of the people in the DRIVE Consortium and my new colleagues at Oxford for a lot of these thoughts. Thank you.
All right, and now we'll move into nutritional information, which David said he was glad he didn't have to talk about, but Walt is going to talk about. So, Walt Willett, value of nutritional information and how to gather it.
Good. Well, thank you, Francis, for gathering us here together to talk about these matters that are very important, and clearly working together, we can do a lot better than we can do on our own. That's one of our, I think, key experiences over the last 40 years that I've had in this area. As David said, diet is, we think, very important for disease etiology, yet it's probably the most complex exposure of all those we've talked about to measure, and a real challenge. Maybe I was just too naive and got into this about 30, 40 years ago, but I think we have managed to learn quite a bit about how to measure diet, and we've learned a lot about that, about the consequences of diet. I'll just cover a few points here. I'll start off going through them. As David mentioned, the high burden of coronary heart disease in most NCDs is due to dietary and other non-genetic factors. Assessment of diet, physical activity, and adiposity is essential in cohort studies, including genomics analyses. Diet studies need to be of long duration for many disease endpoints. Childhood and adolescence exposures may be particularly important. Repeated measures of these exposures is very important. The single best measure of diet is still usually a well-designed food frequency questionnaire. And for weight and height, self-report can be remarkably valid, at least in high-income countries. That may not be universally true. And finally, cohort studies combined with short-term feeding studies will often, short-term feeding studies with risk factors as endpoints will usually provide the best evidence available for translation to policy and recommendations. So I'll try to explain those conclusions as I go along. Again, just to go back to the international correlations done back in the 50s and 60s and 70s, that really essentially energized our work to try to understand the etiologies of cardiovascular disease, cancers, diabetes, and many of the major health outcomes that we're looking at. This is the Keyes famous study looking at 14 populations around the world, and like cancer, there was about a 10-fold variation in risk from the high incidence countries to the low incidence areas. And importantly, the migrants from these low incidence areas like these Japanese villages to the US, which would have been about there at that time, in not too many years, adopted the rates of the new environment here in the United States, saying clearly that these huge differences were not due to genetic factors. There was something about the environment or lifestyle that was important. There were some strong correlations with some aspects of diet, such as saturated fat, but that clearly could have been confounded by lots of other different factors, and this really generated the motivation to try to do more detailed studies and identify factors that may be dietary and other factors that might be causal. I did want to make the point that I know the meeting is mostly about omics, but we really can't maximize our knowledge from the omics without information about diet and some other lifestyle factors as well, but particularly diet. And it relates very much to all of these omic technologies. For example, with genomics, probably one of the most important, strongest genetic risk factors is one we've known about even before GWAS, ApoE4 genotype for dementia, where the relative risk is close to 10 for the homozygous form, and that's itself fairly common. And yet we don't use it, even though we've known about this, and the main reason is that we don't know what to do about it if you have it, and that's not good news just to be told that and sent home by your doctor. But we have now quite strong evidence that diet is very important overall in potentially reducing the risk of dementia and cognitive decline. And we'd really like to be able to address the question whether if you have either the monozygous or heterozygous ApoE4 genotype, whether you will also see the benefits that it looks like are suggested for the overall population. So clearly being able to integrate, as David said, the genetics with our diet and lifestyle factors would be important. And I could talk about all of these, but we're, of course, many of us engaged in microbiome work, very exciting. But one of the questions is, to what extent is the microbiome just the mediating factor for our diets, or are there independent associations? So I think we won't really maximize our knowledge about, useful knowledge about the microbiome without it being integrated with diet. And the same with metabolomics. We're measuring lots of things. Are they direct measures of dietary intake? Are they influences of diet? Or are the things we're measuring due to other variables as well? There have been, over the last several decades, 3 primary methods for measuring diet, and that's been the real challenge. I think we'd all agree we'd like to have good dietary data, but how do we gather that? The classical way has been 24-hour recall, and more recently our colleagues at NCI have developed a web-based version. Some other people have done that as well. The food frequency questionnaire provides a more integrated measure of intake, usually over a year or so. And then another classical method has been the 7-day weighed diet record, which people weigh and record everything they eat for a day, and then it's usually done for a week or 4 days. And so the question has been, which is the best for our epidemiologic studies? And we've recently completed a major validation study of a bit over 600 women in the Nurses' Health Study 1 and 2. And here we also had a unique collection of biomarker measurements that we used, including doubly labeled water. So we could look at the relative validity of these 3 methods. Here, 1 day or 3 or 4 days, 1 average of 2, or 1 week or average of 2 weeks. And the biomarkers themselves all have their errors as well. So what's really useful here is the relative correlation, not the absolute correlation with the biomarkers. But basically the point is that the method with the highest correlation of the biomarkers should be providing the most valid information. This is just a small number of the variables we've looked at, but starting off on the right here, this is 2 weeks of weighed diet record looking at energy-adjusted protein intake. All of these are adjusted for energy. And if you had your choice, you'd take 2 one-week weighed diet records. But the cost of a weighed diet record is about 1,000 times that of a food frequency questionnaire. So it's really, the way we do it, have done it up to now, isn't really possible in large cohort studies. Interestingly, the 2 weeks is only a little bit better than 1 week, and these are the food frequency questionnaires. The 1 week was only a little bit better than the food frequency questionnaire, about the same as 3 or 4 days of 24-hour recalls, and the worst was a single day of 24-hour recall, not surprisingly. And we got about the same relationships with potassium, but different for many other aspects of diet. Here, this is long-chain omega-3 fatty acids, where the food frequency questionnaire was about equal to the 2 weeks of weighed diet records, and even 3 or 4 days of 24-hour recall was substantially inferior. And this it really represents fish intake, I think, as people understand, and that's consumed episodically, so just it's hit or miss on whether you collect the data from the day that you had fish or not. But this represents, well, I think, what we'll see for a lot of individual foods which are eaten episodically. So you need many days of 24-hour recall to get up to what a food frequency questionnaire would do. And this is beta-carotene, and the other carotenoids and many fatty acids look like this too. Very generally, the food frequency questionnaire does about as well as a 1 week weighed diet record, and the 3 or 4 24-hour recalls does substantially less well. So I think that represents, in a very quick way, a lot of evidence that's emerged over the last couple of decades in the dietary assessment area. But one thing to keep in mind, no matter how perfectly, even if you have absolute perfection of measuring diet this week or even this year, that's not going to be perfect information for a long-term follow-up study, because the food supply is changing and people's preferences in diet are changing. So this is just what's happened to intake of 4 major sources of fat in the diets of the men in the health professionals follow-up study from 1990 to 2010. And back in 1990, the consumption of corn oil and soybean oil dominated intakes. And 20 years later, they had dramatically decreased, and olive oil and canola oil had greatly increased. This is maybe a little bit larger change than in the US population as a whole, but the same experience was undergoing in the United States population over time. So you can understand, if you just had that one-time measure of dietary intake by a perfect method, it would have badly measured intake over a long period of time. This is trans fat, and the changes in processing of food have also changed over time. So these are from McDonald's and Burger King. We do these analyses in our own laboratory by gas chromatography, updated about every 4 years. And starting in 1991, in fact, if you'd gone back further, it would have been way up here on the ceiling. But the levels here was 30% in the French fries in 1991, and by 2011 was basically down to effectively 0. So again, unless you have both the dietary intake updated and the food composition databases updated, it's possible to miss a lot that's going on if we're looking at conditions like cancer and cardiovascular disease that develop over decades. So in our large cohort studies, Frank Spicer and Richard Peter were very much involved in getting the Nurses' Health Study going in 1976, mainly focusing on oral contraceptives. And we didn't start collecting dietary data until 1980. But we have been updating our dietary data every 4 years as we go along, and updating our food composition database as we go along in time as well, and using basically the same approach in Nurses' Health Study 2 and our Cohort of Men Health Professionals follow-up study. So we are able to have these repeated measures of diet. I'm not implying that everybody should do it every 4 years. That interval is a little bit arbitrary, but there are huge advantages to having updated information. This is a paper we published last year looking at our combined cohorts, about 130,000 men and women, looking at type of fat and total mortality. There are about 30,000 deaths here, so a lot of power, and type of fat, where we're using the cumulative updated average of all available dietary information up to the start of each 2-year follow-up interval, which gives us the best long-term average intake. And by far, trans fat was most strongly related to risk of total mortality. We've basically seen the same picture in 1997 for coronary heart disease. This is now looking much more powerfully at total mortality. Really same picture. Trans fat by far the most strong. This is saturated fat, all compared to carbohydrate intake. Monounsaturated fat inversely related, and very strongly, this is omega-6 polyunsaturated fat intake related to total mortality. But what if we had just had the baseline dietary data? This is what I just showed you. This is what we would have seen. We would have missed the trans fat association entirely. We would have gotten monounsaturated fat a little bit in the wrong direction. There still would have been a signal, the polyunsaturated fat signal so strong that it would have still been picked up, but we would have missed really almost everything about the type of fat in relation to mortality. Also, and I wanted to make the point that long-term follow-up is important, and some people here have already mentioned that, but I think we're well aware, and those who have been studying cancer, that it's probably some early events occur many decades even before the diagnosis of cancer. And so if diet is going to be important, counteracting the initiating phase stages of cancer, we have to have the dietary measurement there as well. So here we're looking at risk of colorectal cancer using a latency analysis, and the repeated measurements make this much more powerful. And we can see it's not till we get out to over 12 years of follow-up that we start to see significantly lower risk between folate intake and risk of colorectal cancer. And we're doing some updated analyses now. It's suggesting it's really out close to 30 years where we start to see the maximal impact of folate in relation to risk of colorectal cancer. So long-term follow-up is going to be important. And I think that makes all the cohort studies that are out there around the world extremely important, because many of them have decades of follow-up already in the bank that can't be purchased at any price if we started something just today. We can also look at change, as someone had mentioned earlier, as an important part of the evidence, making a case for or against causality. And with repeated measurements, we can look at change in diet. This is looking at change, for example, over 12 years in our Alternative Healthy Eating Index score in relation to total mortality, published last year. And we can see that as well as predicting from baseline benefit, we can also see that change in diet quality is related to a substantially lower risk of total mortality. And then we haven't talked about this much, but in epidemiology, until very recently, we've mainly been looking at how diet and other exposures affect incidence of disease, but there is also a whole set of questions about survival. One of the most common questions someone with cancer asks after they've been diagnosed is, what should I eat? What should I do, doctor? And we've had almost no data to give them any guidance or advice. So with long-term follow-up and repeated measurements, we can look at people for whom we have baseline data, then they get cancer, then we have another assessment of dietary intake, and then we can follow them for mortality. And it takes about 30 or 40 years before we're in that position. And now we are starting to have a flow of papers. For example, this is looking at vegetable fat intake in relation to survival after prostate cancer. Not surprisingly, there's a strong inverse association for total mortality, because a lot of that is coming from cardiovascular disease, but maybe there's some benefit for prostate cancer specific mortality as well, and we'll have to wait for some more data. The idea of working together as consortia is something we've been doing for quite a while. Actually, NCI has sponsored initially our consortia of diet and cancer, the Diet and Cancer Pooling Project, and we now have 35 studies participating in that from around the world. I think a number of people here are participating in this pooling project, and this not only involves putting all the data together in one computer, but involves very active interaction and collaboration among the investigators. And I really want to emphasize, for these complex issues, that's really important. One of the very first papers that came out of this was, in fact, by David Hunter, in fact, the first paper on dietary fat and breast cancer. And this was a very contentious issue at that time, even among the investigators of the cohort studies. And it was tremendously useful to have everybody gather several times, analyze all the data identically for all the cohorts, and whichever way anybody wanted to measure it, the results came out the same. There was basically a null relationship, and that was not only provided very solid data, but it provided a lot of consensus among the investigators, which was important here because this was actually driving a lot of the low-fat dietary advice in the United States, or at least the belief in low-fat diets would prevent breast cancer. Really was not supported by the evidence once we had all the data together. One very important blind spot in the epidemiology of diet and cancer and other diseases is diet during childhood and adolescence. And we're just starting to get data. Now, this is from Nurses' Health Study 2, where we retrospectively asked about, when women were pretty young, about what they were eating as high school students, and we cross-validated that in a couple of different ways. And then we prospectively looked at how that information reported by the adult participants was related to breast cancer. And we do see a different picture than we see about diet and cancer in midlife. It looks like diet is more important during adolescence as we expected. For example, positive association with red meat consumption and inverse relationship with fiber intake. Unfortunately, we can't replicate these findings because nobody else has comparable data. And I would really hope that through efforts in various groups that there would be an effort to be able to replicate this data. Fortunately, our Scandinavian colleagues have now collected data, or are collecting data, their birth cohort, now that they're about 14 or 15 years of age. So in 20 or 30 years, we will have a very good body of data there. But if you don't want to wait that long, we can in younger adults collect data reasonably well about, in retrospect, what people were eating during high school. And then just a very brief word about biomarkers of diet. They can be very useful, but it's not simple because diet, what we measure biochemically is a nutrient in the blood or another in the urine, can represent dietary intake. It can also represent genetic variation. It can also represent the effect of metabolism. And so it's really critical, if we're using a biomarker, to have that in conjunction with dietary intake to be able to interpret those biomarkers adequately. Here, this is a good example. We had seen that if we looked at overall breast cancer, fruits and vegetables were minimally related. If we look at ER-negative breast cancer, there is an inverse relationship with vegetables intake from the pooling project. And then this is looking at another pooling project led by Heather Eliason based on blood levels of beta-carotene and other carotenoids. And for ER-negative breast cancer, quite a strong inverse relationship, and really consistent with the dietary data. Again, we should be careful about the interpretation, and really, as this is a marker of vegetable intake, it might be beta-carotene, it might not be beta-carotene, but something about vegetable consumption is related quite strongly to, inversely to risk of ER-negative breast cancer. Just to reiterate what David said, self-reported weight and height are really quite very good measurements. These are correlations in our cohorts between self-reported weight and measured weight, and these may be better than average, you might think, but this basically same correlation was found in NHANES data as well between reported, self-reported, and measured weight, and height is pretty much the same. And using NHANES data, we found that the correlations with risk factors like blood glucose levels and blood pressures were very similar to using the measured weight and height, just as David had said for mortality. And I just wanted to emphasize again, we have quite a bit of experience as working as consortia. In this NCI consortia, specifically looking at body mass index in relation to total mortality or cancer mortality. We've provided very robust data showing here for cancer mortality as well as total mortality, very strong positive association. And then just to sort of tie things together, we've done several analyses looking at how much major disease could we prevent by a package of lifestyle factors. We did this analysis several years ago, led by Frank Hu. and identified a low-risk group, non-smoking, BMI less than 25, moderate to vigorous exercise, and a diet score, I won't describe, emphasizing type of fat and type of carbohydrate, and modest alcohol consumption. And we could calculate that if everybody in our nurses' health study had adopted that simple set of behaviors, and this does not involve any biomarkers, but that would have prevented 92% of type 2 diabetes. So we know a lot about the prevention of diabetes. For cardiovascular, coronary heart disease, we get about 82% preventable by simple, potentially modifiable diet and lifestyle factors. But only 4.1% of the nurses were in that low-risk group, meaning there's a huge gap between what we know and what we're doing. And that comes to the issue of translation, which has been used quite a bit here. And I do just want to make the point that translation doesn't always mean just a new drug, that it can mean policy and recommendations and behavior change. And so this, just as an example, I think the trans fat story does give us, I think, a good example of how we can use data in a situation where a randomized trial would have been very different to move to a high level of certainty about causality and onto interventions. And these are the data looking at trans fat intake and coronary heart disease risk from our very first paper in 1993. It got tightened up a lot with more follow-up. Here also trans fat related to type 2 diabetes. And then very importantly, there was another line of investigation that were short-term controlled feeding studies of 50 people or so for 2 or 3 weeks given high trans fat versus low trans fat diets. And in those studies, trans fat consistently elevated LDL cholesterol, reduced HDL, and increased inflammatory factors. So putting together those short-term changes in risk factors with the epidemiology, which I should mention was replicated in other studies as well, that combination of evidence it makes a very powerful case for causality. And we then did, with working with some advocates, got trans fat on the food label, and that was motivation for most of the food manufacturers to remove the trans fat from their products once they had to declare it. But the restaurants still didn't stop serving trans fat because there was no food label. They didn't have to declare it. And New York City and Boston and California and a few other places banned trans fat from restaurants, and the levels there came down dramatically in restaurants and in restaurant foods. And we also saw that based on national survey data, a dramatic reduction in trans fat down, I think it's down probably about 95% reduced, and it will be illegal in July, actually, in the United States to use partially hydrogenated fats. The CDC saw a dramatic reduction, about a 2/3 reduction in trans fat levels in blood. It's probably even much substantially reduced today. And in some studies done by economists, actually, there has been a more rapid decline in coronary heart disease in places that adopted restaurant bans than in those that didn't, that were not early adopters of trans fat bans. So I think we can use data that can be collected by relatively inexpensive means to move all the way from our basic measurements to policy and actually start to see some health benefits from that. So I won't repeat the points that I started off with, but just to make the general point that I think the idea of collaborating, working together is tremendously valuable. There is much more to be gained than working just individually here. I do hope that as studies are going to be doing more measures in their biomarkers, they'll also think about collecting updated data on diet, physical activity, smoking, and some of these modifiable risk factors, because that can greatly enhance the data that's going to be emerging from the new omics technologies. Thanks, and I'll turn this over.
Thanks, Walt, very much. Sek, we'll keep going straight away, if you don't mind, and then we'll take a double act to Francis and myself chairing at the end. Sek, it's all yours.
Thank you. Okay, thank you to the organizers for the privilege of speaking today. So my task was to talk about genomic analysis or analysis in ancestries outside of Europe, and I'm going to focus on genetic analyses. And I'm actually going to start by framing it with these two individuals who are clearly of European ancestry, but on the left, of course, is Churchill, and on the right is Jim Fix. And so these two men highlight because of, they both died of cardiovascular disease. Churchill lived to the ripe old age of 90. Fix, unfortunately, had premature coronary disease and died at 52 while jogging in Vermont. And there's a famous quote from Churchill that I think highlights many of the themes that we've talked about today. Apparently, he was meeting with the King of Saudi Arabia shortly after World War II. The king hosted him for dinner. And he, of course, famously liked to drink, smoke, and had a very unhealthy lifestyle. And I think he was advised that the king, really did not serve alcohol, and he could not smoke during the meal. And he's noted as quipping, "My rule of life prescribed as an absolutely sacred right, smoking cigars and also drinking of alcohol before, after, and if need be, during all meals, and in the intervals between them." So he did actually get to drink and smoke during the meal, apparently. And despite all those adverse exposures, again, he lived to a very old age. So these two men, I think, highlight two concepts. One, resistance to disease, and the other, premature disease, basically factors that might actually confer susceptibility, and that's what I want to talk about today. So protection, are there mutations, for example, that might confer resistance, and then why do some get disease at an early age? And I'm going to focus on one disease that we've worked on, and that's namely coronary heart disease, myocardial infarction specifically, and it's a leading cause of death worldwide, heritable, complex disease, interplay of lifestyle and environment, and has two phases to the disease. There's a chronic phase, which involves buildup of atherosclerotic plaque in the coronary arteries, and then an acute phase where plaque disruption, insight to thrombosis, and then if that thrombosis lasts for more than about 20 minutes, the part of the heart that is served by that artery dies, and that death of muscle tissue, that death of muscle is myocardial infarction, can be detected by symptoms, EKG change, and elevation in cardiac biomarkers. So in terms of protection, I think it's useful to think about this schematic. So if you take the population, average genome, average risk of disease, there are some individuals who might carry a mutation, and as a result of carrying the mutation, they actually have lower risk of disease. They're not increased risk, but they're protected. And why might you want to know this? Well, you might want to develop a medicine that actually mimics the mutation and transfer the naturally conferred benefit to the rest of the population. And that's the concept, looking for these so-called protective mutations to guide medicine's development. And there's a specific type of mutation that's actually very useful. These are mutations that completely disrupt protein coding genes, so-called null mutations, and these mutations provide clear direction of effect. Because they either lead to a nonsense mutation, splice site, or indel frame shift, and they decrease a gene's function. And there are two types of scenarios we're often looking for. One is a case where one copy of the protein is gone, so-called heterozygous null situation. And then the second is where both copies of the gene are broken, and it's a homozygous null, or also called a knockout. These heterozygous null conditions can give you insight into the efficacy of potentially downregulating a given gene of interest. And then the homozygous null situation, particularly if the individuals are healthy, can give you insight into the safety of complete inhibition of a given target. So the poster child for this story, of course, is PCSK9, discovered in 2003 by Catherine Boulot and Marianne Abifadel in France, and they found families where there's gain-of-function mutations that increased LDL cholesterol, and these individuals had higher risk for coronary heart disease and stroke. Conversely, Helen Hobbs and Jonathan Cohen identified 1 in 40 African Americans in the United States carried a null mutation in the gene, heterozygous null. This mutation lowered LDL cholesterol lifelong and led to a dramatic lowering of risk of coronary heart disease, risk of heart attack. Now, one important point for this audience, and I think this is underappreciated, is that the mutation that gave key insight here in terms of the direction of effect and actually prompted the medicine's development, that mutation is found only in individuals of African ancestry, only in blacks. So if you did not study this population, you would not actually have this insight that inhibition of this gene would actually be protective. So in addition, African-American woman in Dallas was completely deficient for PCSK9 and was found to be healthy, suggesting that, again, complete inhibition would be tolerated. So this story has now come full circle, where there are two medicines that have been developed to inhibit the circulating protein. These medicines block PCSK9. They've been successfully shown in randomized controlled trials to not only lower LDL cholesterol, but also lower risk for coronary heart disease and overall mortality. Okay, are there more protective null mutation examples? I'm going to give you one story, and this story involves these individuals, founder population, Lancaster Amish. About several hundred individuals from Switzerland emigrated to the United States in the early 1700s. That population has grown to a couple 100,000 people now. And in 2008, Tony Poland noted that about 5% of Amish carried a null mutation in the apolipoprotein C3 gene, or ApoC3. As a result of carrying the mutation, these individuals had lower triglyceride levels. We and a group from Copenhagen showed that actually 1 in 150 in the European ancestry individuals in US and Europe basically carried the same null mutation, and these individuals also had lower triglycerides, but we were able to document there was also lower risk of coronary heart disease in this individual, suggesting, again, that medicines that might antagonize this circulating protein might be therapeutically beneficial. Now, we looked for homozygous null individuals, again, with the PCSK9 analogy, to see if maybe we could find them and would they be healthy, and that would suggest that complete inhibition would be safe. After looking at about 200,000 individuals of European ancestry, we didn't find a single person that was homozygous null. So we turned to an important concept in human genetics, that's consanguinity. When parents are closely related to each other, the children are much more likely to be homozygous by descent at any given site in the genome. This is a map of consanguinity rates around the world. The red, of course, is the deeper shades of red indicate greater proportion of the population are the product of consanguineous unions. And you can see there are particularly high rates in North Africa, parts of southern India, and then in the Middle East and in Afghanistan and Pakistan. The highest rate in the world is in Pakistan, and we established a collaboration with a couple of investigators who had established a cohort in Pakistan to study them for a variety of cardiometabolic diseases, and it was actually led by John Dinesh, who spoke earlier today, and he, along with a student of his, Danish Salihin, really set up this cohort many years ago. We reached out to them to try to think about sequencing this cohort to identify genetic factors for coronary heart disease. And in Pakistan, we identified the world's first ApoC3 homozygous nulls. And this is John, and this is Danish Salihin, who, again, led the study, and this is a postdoc in my group, Pradeep Natarajan. So what you can see here on the x-axis are the three genotypes, heterozygous null, homozygous null. On the y-axis is the blood level of apolipoprotein C3. It's a circulating apolipoprotein, and you can see there are 4 individuals who had no circulating apolipoprotein C3. Here's the larger family for one of the probands, and amazingly there are 15 unions here in this family between first cousins. And we ended up focusing on this individual here, one of the probands that was homozygous null, to really do a deeper investigation in terms of this family. We recruited this nuclear family for hypothesis-driven phenotyping related to the apolipoprotein C3 function. And what you can see here is this is the proband, the mother, 9 children, which is very good for human genetics, and the proband was homozygous null, and to our surprise, the mother was also homozygous null. And so now there are 9 obligate homozygotes in this family, so we went from actually finding 0 knockouts in a 200,000 European ancestry individuals to 9 in just this family. And so we were able to recruit individuals of all 3 genotypes from this family, homozygous null, heterozygous null, and wild type, and to do provocative phenotyping. And so there's been a lot known about apolipoprotein C3 over the years. It's a gene that normally puts a brake on the body's ability to digest dietary fat, and so it's a bad actor. And so we did provocative testing and brought in individuals homozygous null without the mutation and gave them an 80-gram fat challenge. a big milkshake to both groups, and measured every hour for 6 hours afterwards to look at the dietary response, or the response in terms of blood triglycerides to the dietary challenge. This is all led by Danish Salehin, who had a mobile van to do these experiments in Pakistan. So here are the data. On the x-axis is time, on the y-axis is blood triglyceride levels, and what you can see in the wild-type individuals, after a fat challenge, the blood triglycerides rises anywhere from 2 to 3-fold. This is what's happening in us after the meal we just had. But in the homozygous null individuals, there is literally no postprandial rise in blood triglycerides. So with a lifetime of meals, you can imagine that these individuals, their arteries are literally seeing less fat, and so therefore they're protected from coronary heart disease. So now these data suggest that inhibiting apolipoprotein C3 might, again, be therapeutically beneficial. And so there are several companies now that are developing different approaches to antagonize apolipoprotein C3. And these medicines have now been tested in humans to look at triglyceride as a phenotype. And then there are plans to do coronary heart disease outcome trials as well. Now imagine if there was a public resource of such human knockouts from around the world. And this is work that Daniel MacArthur and David Van Heel and a couple of others are trying to put together. And here are a series of papers over the last couple of years that have described human knockouts in several different populations. So this is a study that we did. This is David Van Heel's study from the East London Genes and Health. This is from Decode, and then this is Daniel MacArthur's study from in terms of XAC. And what's interesting is that if you look at the sets of genes that are identified as homozygous null across the 4 different projects, they're non-overlapping. And so you're really going to get a lot of information by studying different groups, because by chance, different genes are basically knocked out in different populations. So it's really going to be important to bring people together, like has been done for these two today and tomorrow, and look across studies. This is a concept that was highlighted earlier. There is some additional value in focusing on populations where there's a lot of consanguinity in terms of getting at homozygous nulls. And so this is the rate that we project with sample size on the x-axis, number of genes knocked out. And in outbred populations, there's a plateau, and that at range of about 150 to 200,000 individuals. But if you focus on a population like PROMISE, the Pakistani cohort, where the inbreeding coefficient is a bit higher, then we estimate that with about 200,000 Pakistani sequence, you'd identify about 8,000 genes that are in the homozygous null state in at least one participant. In addition, I think India is a part of the world that is particularly, this kind of genetic research may be quite fruitful, and this is because of work of David Reich and a couple of others that have shown that founder populations like Ashkenazi Jews and Finns, and also the Amish that I mentioned, in India there are 14 groups of more than 1 million size with founder events more extreme than those 3 populations that I just mentioned. And so this is something that really we should think about as we move forward, how to kind of tap into this set of this population. Okay, in the last few minutes, I'll just go over this idea of risk, the flip side of the coin. And if we think about why some develop MI at a very early age, like Jim Fisk, there are several models, of course. Monogenic model, single mutation sufficient to drive the patient to early MI. Polygenic model, the additive effect of many variants leading to early disease. And of course, lifestyle, as highlighted by Drs. Hunter and Willett. So I'm going to focus on the polygenic component here. If you think about traditional approach to prediction and thinking about genetic factors for disease, we often focus on rare monogenic mutations in terms of genetic prediction. And if you think about rare monogenic mutations and risk for heart attack, the main contributor is familial hypercholesterolemia. These are mutations in LDL receptor, ApoB, or PCSK9, increased LDL cholesterol, and the individuals have about a 3-fold increased risk for myocardial infarction. So but this model, the monogenic model, is applicable in a very small proportion of the population, about 0.4% of the U.S. population. So we wondered whether can we identify additional patients with a polygenic risk model. And the typical concept over the years has been taking n number of polymorphisms that exceed a genome-wide threshold in a GWAS study, 0, 1, or 2 copies of the risk allele, and then a score ranging from 0 to 2n for each person. And so this is the typical way to kind of do these polygenic scores, looking at basically the top of the list. But we wondered whether if you actually incorporated information for the rest of the genome, from the rest of the genome, could you actually add predictive power? So we moved from just the top SNPs in terms of polygenic risk prediction to for coronary disease, actually including 6.6 million variants in a linear combination in terms of scores, weighted scores. And this is work that's up on bioRxiv as of late last year, led by Amit Khera in the lab. And so here's the study design. So we took results from a published genome-wide association study for coronary disease, tested different scores in the first phase of UK Biobank, and then validated, picked the best score here, and then validated that score in the remaining portion of UK Biobank. Now, one important point here is these genotypes, for the 6.6 million variants, do not involve whole genome sequencing. It's basically common variants from the genotyping array that's been done in UK Biobank, as well as imputation. So here's the score. So it's a beautiful bell curve, basically a new quantitative metric of genetic liability to heart attack, standardized to a mean of 0, standard deviation of 1. So this is the distribution in the validation set. And so this is very similar to other traits like LDL cholesterol or height or blood pressure. So we wondered, what if you happen to be in the extreme end of this bell curve? What is your risk compared to everybody else? Could it actually approach that of a monogenic carrier status for coronary heart disease? Before I get to that answer, we also looked at the correlation of this score on the x-axis. On the y-axis is the Framingham risk score, or updated to be now the ACC-AHA pooled cohort equations of traditional risk factors. And what you can see is this genome-wide polygenic score has very little correlation with currently measured MI risk factors. This is a correlation coefficient right here. So this is actually capturing something completely orthogonal to what we use in clinical practice right now. So here's the data on labeling the top 5% of the individuals in this score as carriers and looking at their risk compared to the remainder of the distribution. And it turns out their risk approaches, or actually greater than, that conferred by a monogenic mutation. So the odds ratio here is 3.3. If you go out to a little further on the tail, the top 1% of the population, the hazard ratio is almost 5. So this is a comparison then of the monogenic and polygenic contributors to early MI. So the monogenic LDL receptor, PCSK9, ApoB mutations present in 0.4% of the population. Here's the odds ratio for MI. These individuals can be found by genetics, but also can be found by the fact that they have higher LDL cholesterol. And the mechanism of risk here is ApoB containing lipoproteins. For polygenic, this at the 5% tail, the individuals have a threefold increased risk as well. They're currently unaware of their risk. These individuals are not distinguished based on their traditional risk factors, as I showed you earlier. And then, for lack of a better term, the mechanism here is really truly a gemisch. It's additive effect of many, many variants. Now, what about intervention? We know that lifestyle and medications can modify the LDL and modify the risk of Mendelian forms of MI. What about for polygenic risk? And so we have looked at this, and I think David highlighted our recent work showing that adherence to a favorable lifestyle, even if you have high genetic risk, is associated with lower risk of coronary events, incident events. And then we've also shown that individuals who have high genetic risk, high polygenic risk, actually benefit quite a bit from statin medications. Okay, so here's the summary then of monogenic, polygenic, and combined. If you take 100 individuals with early MI, about 2, the risk is due to monogenic basis. About 15 to 20, it's polygenic, and then there's some unfortunate souls who have both, and it's additive. So just in the last 2 minutes then, this approach that we've now developed for a genome-wide polygenic score is relevant not just for coronary disease, but we've been able to show that it's relevant for a range of other common diseases: atrial fibrillation, diabetes, inflammatory bowel disease, breast cancer, and obesity. And we find the same pattern that people at the top end of the distribution, the tail of the distribution, basically have much higher prevalence of disease compared to those at the lower end. And these dots are 1% bins. So why is there much better prediction now compared to earlier? And that's because of larger genome-wide association studies, better methods to bring together genome-wide scores, and then lastly, large cohorts like UK Biobank, where you can test and validate to find the extreme tails at higher risk. So now, will the MI polygenic score that we derived and validated in individuals of European ancestry be generalizable to other ethnicities? So all three components here are basically individuals of European ancestry. So this is a big question. So we looked at this, and it does generalize. So this is data now of that same score, the top 5% tail, and the hazard ratio in whites, blacks, Asians, Hispanics, and South Asians. And you can see here for whites, it performs the best. In this study, about 4-fold higher risk. And it does relate to risk of coronary disease in the other ethnicities, but the effect estimates are attenuated, largely dependent on how far away they are from Europe in terms of ancestry. So this really now emphasizes that it's important to expand this approach, polygenic risk scoring, to other ancestral backgrounds, including maybe even doing these GWAS studies in non-European ethnic groups. And that exposes actually a big challenge, which is only less than 20% of the published genome-wide association studies are individuals outside of Europe, ancestry outside of Europe. It's gotten better since 2009, but really the non-European ancestry groups are lagging behind. Okay, so multi-ethnic studies and specifically focusing on genetics, the opportunity is that gene variants providing unique biologic insights are distributed all over the world, and we really do need to look at the full range of diversity, genetic diversity, to get at unique biologic insights. And then the challenge is the way the setup is right now, the polygenic scores are really optimal, going to be best constructed in individuals of European ancestry, and I worry that those of non-European ancestry could be left behind, and that's a situation we need to rectify. Thank you.
So we are going to let the discussion go an extra 5 minutes. So we've got till 3:15, according to our distinguished leaders here, for discussion. So let's open that up. Maybe I'll start while people are trying to decide who wants to raise their hand. I want to ask Walt. It's actually surprising to me that somehow the questionnaire model still seems to be, of the things that were assessed, the most reliable, because it seems like it is a measure of dietary intake that people love to hate because of all of its shortcomings. And I guess one has to wonder, nurses, aren't they more likely to be a lot more careful about these things than the average person? Can you really extrapolate from the Nurses' Health Study the reliability of that kind of questionnaire? And if we're going forward and everybody has a cell phone, is this really the way we want to do this going into the next decade or so in terms of collecting this critical information about dietary intake?
You did ask a couple questions there. The first one, are the results generalizable to other populations? And yes, the other people have conducted similar validation studies and get results very similar to what we did. There's nothing very sophisticated in those questions that requires medical training or anything like that. Obviously, you do have to have some motivation and commitment, but basically, other people find very similar results. And is there something better? There's quite a bit of work being done now trying to see if using cell phone pictures and other kinds of methods might be better. I think it's fair to say this has been looked at for quite a while now, and so far no one's ever shown that they can do better. But as the new methods are being developed, they really need to be rigorously tested, sort of in parallel to the kinds of approaches that I just showed, comparing that new method with another method. Partly, I think, despite what, as David sort of alluded to, there is a little bit of prejudice against people being able to describe what their own behavior and their choices. But in fact, people do it reasonably well. And I think that 2 weeks of weight diet records is probably about a ceiling of what we can get. There's no memory involved there. People are doing it in real time, weighing and measuring. And the food frequency questionnaire is not very far behind that. So I think the margin for improvement, it's not, yeah, I'm sure we can do fine tune and enhance things a little bit more, but there's not a huge gap there between what seems to be a ceiling and where we are now. That said, different aspects of diet are measured differentially well. The things like contaminants we don't measure very well. Biomarkers are going to be especially helpful there. Things that are naturally the constituents of food, we will do quite a bit better. So that we need a variety of approaches. Now, there's not one single approach that will be optimal for all aspects of diet. One of the reasons that the cell phone sort of picture is limited is that can't tell a Diet Coke from a regular Coke, or what's inside a wrap. Is it beans or is it red meat? So you need to add some other information, some sort of narrative to go along with it. I wish we were like physical activity where this accelerometer, you just put it on and forget about it. It does pretty well. I don't think we have anything or are likely to have anything that is analogous to that for diet.
But innovation is a really great thing, and if there are bottlenecks to being able to improve critically important parts of what we're doing, it would be good to identify those, and then people in this room or beyond might take that on as a critical step to change, even though it may seem difficult at the moment. Move on. Other questions or comments?
Yeah, this is for Dr. Kathiresan. My bias is
I'm not sure the mic is on.
well, I'm doing all I can do. Now it should work. Okay, great. So my bias is I'm a clinical geneticist by training, and I'll preface my remarks by saying that I agree with you that the combined approach to identify risk for myocardial infarction for monogenic and polygenic is the way to go. And I'm pretty sure you didn't mean to say what you said, but the point that you raised about the fact that monogenic forms of hypercholesterolemia can be identified by LDL cholesterol levels actually tends to reduce the fact that there's an independent risk associated with having a pathogenic variant in LDLR or PCSK9 or ApoB that essentially doubles the risk independent of your LDL cholesterol level. So if you have a low LDL cholesterol level, you still have twice the risk if you carry a variant as opposed to an individual that does not. Now, what we don't know whether that's the case for polygenic at the present time, whether that represents an independent risk or whether LDL cholesterol is a good surrogate for that risk. So I think we need to look at both of those questions in both of those conditions, but to focus on one or the other, which I know is not what you're advocating for, would have its own set of risks.
Yeah, actually, we were the ones who made the observation regarding the additive role, the incremental value of knowing the genotype beyond the LDL cholesterol. But even with that, it's still, in the United States, it's the current practice right now is to screen based on LDL cholesterol, which is actually, there's a pretty good argument to be made for both, either the genetics and or the LDL. Because, for example, if you had a person with a mutation and their LDL is low, you wouldn't go treat that patient because it works through the LDL. But I don't think it's an either or. I think my main point was only about 2% of patients who have early MI is there a monogenic component. But there's a huge role of inheritance for early MI. Where is the rest of the inheritance coming from? It really looks like it's coming from a polygenic model.
Just whilst on that multi-ethnic, and just for those who don't know, the NIH and we are working very closely together on something called H3 Africa, which is not cohort in its design, but nevertheless is trying to shed light on the genetic variability in Africa. And H3 Africa at some point should be brought into this discussion going forward.
75,000 samples. Oh, yeah, Nikki's here. H3 Africa is here, yes. Hi, Dan.
Others?
Maybe everybody in the room got it except me, but I don't think you said specifically that the genetic risk score can also be used, I think, as a predictor of penetrance of monogenic diseases. So one of the things that we find in, and I was really distressed that the QT interval wasn't on your list of phenotypes where the genetic risk score has shown value, because it has. So you can add that to your list. But the places where that may be important is in managing families, where we run into this problem of, a variant carrier has an extreme phenotype like dropping dead, and then there are other variant carriers in the family, and you're not quite sure what to do with them, and that may help.
I think it's a great point, and I think I want to emphasize the area that we're very excited about, in addition to some things you mentioned, is actually breast cancer. So the majority of women who get tested for, because let's say they have familial breast cancer, the majority of them do not carry a BRCA1 or 2 mutation, so less than 1 in 500 would. And but the polygenic risk score for breast cancer, if you're in the top tail, let's say 1% tail of the distribution, you're basically approaching levels of risk equivalent to the BRCA1 or 2 mutations. And so they could basically modify risk in BRCA1 carriers, but also in non-carriers, it's an important contributor.
John.
Question for Sek. Smashing talk, thanks very much. I'm behind you, Sek, in case you're wondering. Yeah. So two things. One is, could you, for my education, could you tell me with the human knockouts that you and Pradeep did the analysis on and discovered in the Promise paper, that were not expected to be able to be compatible with life based on developmental biology work in other species? Could you comment on your reflections now, kind of a year later, why you think we believed what we did before and then discovered this in humans? Why was there this mismatch in understanding?
In both our work and also that from David, and maybe David Van Heel can speak to this as well, because they found a very interesting knockout for a gene PRDM9, which was thought to be incredibly important in terms of knockout incompatible with life, but they found a person who did carry it. The few genes that we looked at, one of them, for example, Titin, and there's a Titin knockout, and with a normal echo, living to adulthood, and in a part of Titin that was thought to be important. It's not a misannotation, for example. And exactly why, I have no idea. I mean, I think it's really, clearly there's a lot we don't understand about even a gene like Titin, which has been heavily studied.
And as a follow-on, if I could just ask, so I wonder if whether you or Daniel MacArthur would like to reflect on, you introduced the exciting concept of a human knockout project, on what you think might be, given some of the discussions today about novel types of molecular phenotyping, et cetera, what you think might be the first pass triage to try to work out what are some of the phenotypic consequences of a catalog of human knockouts, and how you would propose to characterize knockouts?
Yeah, I think ideally what you like to do is find these individuals and then have a standardized protocol where you would evaluate them similar to the way the model organism work is going, where you essentially understand a gene's function by getting a mouse knockout, for example, and those knockouts are phenotyped on a battery of tests, standardized. And ideally, that's what we'd like to think about doing, and it's going to require thinking through what those tests are, where these can be done. In some parts of the world where this is more prevalent, like Pakistan, what's available in terms of resources. And so I think it's worthy of additional discussion.
Jeff.
This question is for David. At the end of your talk, you mentioned a study that is aimed at early disease detection. So I'm gathering that that's just not a study where you just do get a sample and then do longitudinal follow-up, that there's a much different sampling strategy in order to actually detect disease at an early time point. I'm sorry if I missed this, but could you comment on elements of study design if you really want to do early disease detection?
Yeah, so this is actually something that's of real interest to Cancer Research UK, and they talked with Biobank, but as I'm sure you know, to protect the circulating tumor DNA and not have it swamped by genomic DNA, you have to use special tubes. And so most of our biobanks aren't able to address this question about whether through next-gen sequencing you can pick up cancers 1 or 2 years prior to clinical diagnosis. So the sort of bullseye I showed, I think CRUK is very interested in setting up another blood cohort, and I'm trying to persuade them at very low marginal cost to enroll people in a questionnaire only or a spit only who don't choose to give a blood sample, because that's a hazard. The other option is to sort of do it the other way, outside in, and get people in a study and then ask them if they want to be in these studies. There's obviously other ways you could do this. Follow up people who are at high monogenic risk, but they tend to be heavily surveilled. So hopefully, anything they have will be picked up very early, or in the case of something like HNPCC, they'll have their polyp removed, so they may not be informative. BRCA1, obviously, women may have prophylactic oophorectomy or prophylactic mastectomy and sort of take them out of the risk pool. So our calculations are that to do this, you would need a very big biobank to have adequate numbers in the first year or two of incident cancers. And then, of course, you would have a biobank for all of the other things that you want biobanks for.
So we're running short, but David, and
David
then
Van
I
Heel.
have one quick question for David Hunter.
Can I respond to a previous point and then ask you a new question, actually? On the human knockouts, we've had decades of genetic research which has been looking at rare pediatric diseases, and that's where the human knockouts have been found. And only until very recently has anyone studied adults whose parents are first cousins, who are a reasonably healthy population, and surprise, surprise, they have knockouts too. I mean, both have their ascertainment bias, and one way you're going from extreme phenotype and finding the genotype, and the studies that we've done are finding the extreme genotype and then looking for other things. And I've got another question for you, which is on the polygenic risk scores, and there's a lot of controversy at the moment that a lot of polygenic risk scores pick up variation in ethnicity, and some people